Lecture 2 is going to be all about muscle physiology. Because this is kind of a big topic, this will be split into two different sections. So this is Lecture 2a, and we left off in the last lecture talking about muscle fibers. So that's where we are here. This is the muscle fiber, the muscle cell. Inside the muscle fiber, we have these bundles of proteins. Those are called myofibrils. So myofibrils are organelles inside the muscle cell that are made up of bundles of protein filaments. Notice that the myofibril is stripy, so we can see that there are dark regions and light regions, and that they repeat in a really regular pattern. This patterning of the myofibril is what gives skeletal muscle its striated appearance under a microscope. When we look at the striations of a myofibril, we can see that there are light bands, which are known as I bands, and dark bands, which are known as A bands. The I bands have what's called a Z line or a Z disc right down the center, and we see this label sarcomere. A sarcomere begins at one Z line and ends at the next Z line. We'll talk more about those terms. They'll make a little bit more sense later. This picture again starts with the muscle fiber. We can see that the muscle fiber is filled with these myofibril protein bundles. And if we look more closely at a myofibril, we can see that it is striated. There are dark bands and light bands. The dark bands are the A bands, light bands are the I bands, and we have Z discs in the light bands. If we look even closer at just one little section of a myofibril, we can see what causes the light and dark bands. So down here, notice that we have overlapping filaments. The two types of filaments contained in the myofibril are actin, which are thin filaments, and myosin, which are thick filaments. In this picture, the myosin is the red and the actin is the light blue. And those fibers overlap in a regular pattern to create the striations. The A bands are going to be seen anywhere that you have myosin. So the thick filament is what creates the dark band under a microscope. I bands are everywhere that you have no myosin. So where there are only thin bands, you see a light stripe under a microscope. Couple more vocab terms here. So again, remember that there's a Z line in the middle of every light band. Basically, the Z line defines the end of the sarcomere. So this is where one sarcomere ends and the next begins. The thin filaments, the actin filaments, are attached to the Z line. So in this picture, everything that's red is actin. The sarcomere in this picture goes from this Z line over to the next Z line. And the sarcomere is the contractile unit of the muscle. So it's going to be the part of the muscle that actually shortens. The H zone is not labeled in this picture, but the H zone would be everywhere that we have only myosin. So right here across this middle area. The M line is right in the middle of the H zone. M line is an imaginary line. There's nothing physically there. It's just sort of marking the middle point of the sarcomere. So the middle of the sarcomere has this empty zone where we have only myosin, and we just mark the middle of that as the M line. We'll look more closely at how sarcomeres contract, but basically the H zone is important because it's the part that's going to shorten. It's going to disappear as the muscle contracts. So a fully contracted sarcomere will not have this empty space between the actins. The actin will be pulled all the way in towards the M line on both sides. This is just a really good recap picture showing you all the way from the muscle as the organ to the tissue. So here we have a bundle of muscle fibers called a fascicle. If we look just at one muscle fiber, we can see that it's filled with the myofibrils. If we look closely at the myofibril, we can see that there are A and I bands, And if we zoom in even closer to just a couple of protein fibers in that myofibril, we can see that the A bands are caused by this thick myosin filament, and the I bands are caused by the lack of thick myosin filament. I bands are where you have only thin actin filament. If we look even a little bit more closely at each of those two filaments, here's what they look like in a little more detail. The actin filament are two very, very thin chains of protein twisted together, whereas the myosin filament is much thicker, lots and lots of smaller pieces of protein twisted together. Here's a quiz for you. Pause the video if you want to stop and try to fill these in. I will tell you the answers very soon. Okay, so here you can check your answers, and we're going to move on. Here's one more view of the same thing. Take these vocab terms, fill them in on the picture. This list of vocab terms actually also includes some of the connective tissue layers from the first lecture. Pause the video if you want to work on this, and check your answers. So I mentioned that sarcomeres are the contractile unit of a muscle. Here's how that looks. So here we have two different pictures of a sarcomere. This is a relaxed sarcomere, and this is a contracted sarcomere. The difference that you can see is that in the relaxed sarcomere we have an H zone. So we have this area where the myosin is bare and the actin filaments, the light blue, are not stretching all the way in towards the center. Down here in our contracted filament, there's no more H zone. The actin goes all the way in towards the center and overlaps. Continue to look at the explanation on this slide. There's another picture on the next slide that I'm going to move ahead to. So again, here we can see a relaxed bit of a sarcomere and a contracted bit. The relaxed sarcomere has a good deal of space here between the Z disc and the myosin, and it has a good deal of space in the middle here where the actin filaments are not touching and where we have only H zone, we have only bare myosin. A muscle contraction occurs when these little heads of the myosin protein reach out and grab onto our actin filament, and then the myosin pulls on the actin and pulls it inwards towards the center of the sarcomere. So this is the relaxed is the before, contracted is the after a pull has occurred. The next thing that the myosin would do would be to let go, grab on a little bit farther down, and pull the actin inwards again until the actin is all the way in towards the center. Okay, here we have a quick video. This is showing a single sarcomere, and it's going to show the contraction. Just a quick rundown. We have myosin in the middle, this thick sort of tan structure. Our actin is this reddish thin structure, and we've got these purple Z discs. When the sarcomere contracts, the Z discs pull inwards and the actin meets up in the middle. Now remember that the sarcomere is just one contractile unit within a muscle fiber, and the myofilament is made up of one sarcomere after another, and they're going to be all shortening together at the same time. Okay, that's it for this segment of the video. The second part of this lecture will talk about how the filaments actually slide. So how does the myosin actually grab onto the actin and pull it inwards? Stay tuned for that one. Don't forget too, there's a sliding filament tutorial online as part of your unit plan that you can do as well if you'd like some extra help with this.